Hello everyone, I'm Dr. Vanati Perumal, the upcoming CEO of Medonc Therapeutics. Today, I'm here to discuss about the live CTC sphere blood testing as an improvement to the existing technologies for cancer therapy predictions and drug development. We are based in Perth, Western Australia, as a medical device small business model. My disclosures are Sir Charles Gardner Hospital Radiation Oncology Fellowship, Curtin University Med School Research Consumable Funding, and Australian Technology Network University's Sydney Commercialization Training Workshop Travel Grant. We are excited in translating our world's first NHMRC proof of concept circulating tumor cell diagnostic products to clinics for radiotherapy, radiochemotherapy kinetics, and other cancer therapies. As shown in the figure, introducing the current cancer status circulating cancer stem cell sphere testing will help move the Australian SFRP4 patent peer-reviewed publications for a quick therapeutic clinical goal, particularly for Winter's cancer patients. Our international team in India is working on SFRP4 small molecules. To simplify and accelerate the drug development process, we are aiming to develop patient cancer spheres for SFRP4 treatment, different to conventional therapies. Well, it might sound ambitious but sensible and achievable approach for novel breakthroughs with good funding, budget, and decent timeline projects. Step 1, culturing SFRP4 upregulated overexpressed patient spheres, and in step 2, scaling up patient cells outside the patient to test for the drugs, example SFRP4 and WINS, which are working will reduce the cost and toxicity than the one-size-fits-all therapies. Purifying human SFRP4 proteins, then the commercial available ones, will help to treat SFRP4 downregulated patients directly at the clinics. Worldwide, around 18.1 million people are diagnosed of cancer. Every year, 10 million people die of cancer according to WHO report in 2020. The common cancer deaths were due to lung, colon, rectum, liver, stomach, and breast. Countries lacking the screening programs are detected at the advanced stage. 70% of deaths from cancer occur in low and middle income countries without comprehensive treatment facility. Survival rate can be improved if the cancers are diagnosed at the earlier stage with the appropriate screening program, particularly where we cannot access or the cancer is in a risky location, avoiding the complicated invasive procedure by a simple blood testing. Tissue biopsies CSC does not reflect the current cancer genomic signature status. It keeps changing with treatments. To help doctors better understand the cancer before symptoms appear or even tumor mass forms, that is, before patient radiographic imaging progressions, ctDNA and cfDNA detections are not at the cellular level, and they are after risky internal drug treatments exposing to drug toxicity, side effects, and with unwanted cost. To tackle recurrence, reducing more invasive and painful procedures on different spot biopsies, we need to introduce simple blood testing to assess the treatment process faster with more accuracy and sensitivity. Most importantly, it will help to understand the whole body tumor heterogeneity in a simple blood tube for more effective treatments. These are my PhD and part-time research publications credentials from Curtin University with full funded Curtin International Research Scholarship and stipend through national and international collaborations. What do we need to do for technology improvement in the clinical space within our research capacity? Only very few skillful labs technologies cultured viable CTC for drug discovery in the world. Most labs focused on CTC detections without larger patient cohort statistical significance, which limits their commercialization prospects. At Medonc Therapeutics, follow up to our previous discovery, as shown in the figure, we wish to introduce the new generation CTC sphere SFRP4 Wind signaling treatments to patients. With the SFRP4 Wind signaling and cancer stem cell background, we established viable CTC and spheres culturing for radiotherapy prediction, sensitization, and drug discoveries as highlighted here. In brief, we are the first group to report on viable CTC lines and tumor sphere clonal cells blood testing for Wnt signaling inhibition drug discovery development platforms. Additionally, it aimed at circulating cancer stem cell responders and non responders predictions. Such interventional clinical trials will improve the quality of life by escaping the internal toxicity and side effects. Besides, it will assist in precise drug selection management by reducing the cost of ineffective drugs and toxicity. Previous studies have shown SFRP4 are downregulated in mesothelioma tissue and cell lines. In addition, we also demonstrated promoter methylation of SFRP4 in GBM models. Our laboratory has previously shown downregulation of SFRP4 in mesothelioma cell lines. We have also investigated the vesicular trafficking of SFRP4 and observed that the neutron-like domain has a vesicular association signal, and most significantly, intracellular redistribution of SFRP4 was observed in response to Wnt3a, suggesting Wnt3a can modulate intracellular localization and secretion of SFRP4. In continuation to my PhD finding, we collaborated with Oxford University to study the function of SFRP4 domains and mutant proteins on cancer stem cells. GBM showed significant effect on NLD domain treatment, and these are unpublished data. We further examined CD133 and ALDH expression in JU77 and ONE58 suspension malignant mesothelioma stem cells. We continued looking at the effect of SFRP4 and its peptides alone and in combination with cisplatin, reduced cell proliferation and disrupted tumor sphere formation. In conclusion, we thus identified for the first time that SFRP4 and its associated peptides could help to sensitize and destroy tumor-initiating stem cells derived from two mesothelioma cell lines and may lead to the development of an effective treatment regimen to combat malignant mesothelioma. As a new therapeutics, future studies will examine the effects of our peptides using human CTC spheres therapeutics with the view of implementing their use in the clinical blood testing setting. Our mission is same as Vortex Microchip Intact Live Cell Technologies, introducing live CTC technologies for understanding the CTC biology and drug development will have huge commercial potential. Our technology could be automated and scaled up to 50 mL for patient circulating cancer cell drug development to support microfluidic chip device and scale up volume limitations. We need more improved technologies to capture all types of CTCs intact in larger volume for patient-specific cell drug manipulation, and delivery. And this technology could be also applied to other disease model settings for genetic testing in a prenatal setting and blood stem cell therapies. Open questions. What role does SFRP4 circulating cancer stem cell therapy play in radiotherapy, radiographic imaging, chemotherapy, and other anti cancer therapies, early cancer detections, including coronavirus cancer patients, for monitoring resistance and sensitivity? Commercially available technologies provides the combination of dead and live cancer cells, mostly focused on metastatic cancers. Improvised to the existing CTC technology models as an inventor in bridging the gap of CTC and SFRP4 drug development, developed the proof of concept products for head and neck cancer radiotherapy feasibility study in compliance with NHMRC and WA health infrastructure. Methods are embarked for trial completion and patent development. The pilot trial products are viable head and neck cancer CTC counts with high recovery, specificity, and purity for radiotherapy predictions, and viable head and neck cancer circulating cancer stem cells sphere count with high purity in 18 hours faster turnaround time, and viable CTC line and sphere culturing for early cancer detection as incidental finding. Further to our previous mesothelioma cancer stem cell peptides finding, introduced viable circulating tumor cell SFRP4 and cisplatin combination treatment in advancing to previous tissue cancer stem cell publications and expired patent products. Interesting cancer risk cluster dissociation effect was noted with limited cultured cells similar to a Cell paper Swiss group on FDA drug screening and mouse model. In this pandemic, for a future sustainable economy, there is still a commercialization scope for 150 billion USD market need for other solid tumor detections, early cancer screening and prevention, and cancer stem cell diagnostics. I will share some interesting finding for future collaborations and expansion to bring quality product to the market as high standard labs, quality publication data. We used appropriate positive and negative control, experimental controls for organ specific cell lines with statistical significance for a longitudinal follow-up study. Here we used 3 spiked head and neck cancer cell line for measuring patient metastatic sphere culturing potential with 98% recovery, sensitivity, and purity on spiked head and neck cancer cell for radiotherapy treatment. As a single step faster turnaround technology, our lab based test allowed to quantify the number of viable tumor cells in the blood as circulating cancer stem cells. The methods were also simplified to multi step CTC enrichment, and we were also able to cryopreserve, revive viable solid tumor morphology, cancer associated fibroblast like circulating tumor cell lines. Additionally, we were able to generate numerous primary and secondary spheres from the same wells. Surprisingly, during validation as incidental discovery, 5 volunteers out of 13 negative control groups yielded 6 to 10 days of viable CTC lines. On cryopreservation revival, the viable CTC lines were like solid tumor cancer associated fibroblast. The proof of concept study was on a 22 to 48 years old healthy volunteer group, detected viable CTC lines as 38.5% for risk and 61.5% free from cancer. In supporting this finding, recent very small embryonic stem cell OC4 group from India and Singapore with statistical significance published their finding in stem cell reviews and reports on known cancer groups. Our products are with additional drug testing solutions, live cell immunostaining and genomics, comparing to known cancer tissue organ signals, measuring the tumorigenicity on appropriate animal models for identifying the cancer type will open doors to new interesting early cancer detection and prevention drug therapeutics. Required more organ cell type or inflammatory markers, signals, experiments for a follow-up care, and measures for early cancer screening and follow-up care for these cancer risk patients were limited with funding and ethics approval from a seed funding perspective. Real false positive and negative controls percentage should be taken into consideration. This interesting incidental finding is noted for future research direction. To offer a solution to current treatment challenges within our research capacity, we introduced SFRP4 treatment in short-term CTCs for complete elimination of cancer stem cells and cancer relapse approaches. New dose and cell model innovations was introduced unique to my own previous published SFRP4 concentration on equivalent spheres with limited CTCs. Successfully generated numerous primary and secondary tumor spheres from spiked CTC lines for SFRP4 when targeted preventive drug screening alternatives. Sensitization and anti-cancer therapy follow-up approaches. As expected, SFRP4 dissociated the cancer risk aggregates. The feasibility study allowed us to provide custom-made circulating cancer stem cell blood testing services to build better patient representative drug testing for empowering cancer research and cancer care from head and neck cancer cell lines and healthy volunteers. The outcome of my talk with global initiatives will set the stage for external investor funding and industry partnership opportunities for advanced cutting-edge technologies. Measures introducing routine live CTC blood testing and treatments for WA community as an occupational health hazard measures for asbestos-induced mesothelioma, head and neck cancers, breast and gastric cancers, and other solid tumors through collaborations. Finally, I would like to thank Dr. Josha Das and the team for commercial project, mindset training, mentorship, grant writing, funding, and clinical trial opportunities. I also thank Sir Charles Gardner Hospital, Curtin University, and Australian Government Immigration for supporting international research innovation opportunities. I also thank Professor Arun Dharmarajan from India, Sri Ramachandra, for the mentorship and international grant writing opportunities. Now, I would also like to thank United Pharma, US, Hanson Wade, Archdex, and Merck for grant success opportunities and invitations. Thank you. I'm open to collaborations.